why don't we just start with the first one from Luigi Notarangelo. Although endothelial cells may be the target of ADA2 deficiency in strokes, I wonder whether the culprit is microglia function, which would be consistent with expression in myeloid cells. Has anybody looked at microglial cells in these patients? And also, do normal microglial cells, in fact, express and secrete ADA2? So that is the question. And is anyone on the stage prepared to answer that question? So Paul, take it away.
I think that's a great question, and the part of the answer is I don't know of anyone who's begun to look at microglia or other neurological cells yet, but the reason I think it's a really exciting question is that we have at least 3 patients in our series who have an evolving neurological phenotype without any evidence of a vascular insult to explain that. In other words, is this enzyme actually doing something within the brain, the nerve cells of the brain, and other associated cells, irrespective of whether there's an ischemic insult to cause the neurological clinical phenotype?
Mm-hmm. Okay. Anyone else like to comment on that question? I think it's an extremely important question, and I think that it does underscore the point that, in fact, some of the early studies that we and others did looking at the cellular locus of expression of this gene, it's turning out that there is a broader spectrum of cell types that are involved, and we need to look at that. Mike?
I mean, we haven't done it, but have you or anybody looked at CSF for enzyme activity? I mean, we'd be happy to do it if anybody has samples stored or something.
Yeah. So we have not looked at CSF for enzymatic activity. Does anyone else? Certainly that would be a related question and an important one.
But it's definitely been sampled in other lesional tissues, so there's some data on, I think, tuberculosis. When you look at pleural fluid, you can measure ADA2 levels. So yeah, we should start looking at different compartments.
Mm-hmm. Yeah, and of course another angle on this is the question of, because some of these tissues are relatively inaccessible or would be difficult to obtain on human subjects, is to develop an animal model. And of course the animal model that our group had developed, at least in the initial New England Journal paper, is a zebrafish model, which although it was interesting and perhaps informative in terms of some aspects of neurologic development and CNS development, it doesn't lend itself to the kinds of studies that one would like to do for this kind of question.
To Paul, how was the systemic inflammation in those patients with just strokes and so on? I mean, like in primary SNS vasculitis, which there is no sign of inflammation, or there is a sign of inflammation?
So are you asking about all of the patients with neurology, or the ones just with the neuro? So they have completely normal inflammatory markers, completely normal. And the signs are, that the first alerted me to, is about 30 beats of ankylosing on both sides. And this boy now in his mid-20s, and the younger sister is about 6, and she's beginning to develop that as well. And we've scanned their brains and their cords, and they're completely normal. And their serum amyloid A and their C-reactive protein and their ESR, platelet count, white count, all the things that we like to test clinically are completely normal. So the question is, are they having small strokes that you can't just detect on MRI, or, coming back to the original point, is it doing something at the nerve cell level, actually?
Can I add something to that? Sorry, Susan.
Yes, Susan.
So, sorry to add something. We have a large group of patients with primary and secondary CNS vasculitides, and I think it's really important to separate out vasculitic strokes from vascular fragility strokes. So kids that the most common childhood stroke is vessel wall inflammation, which leads to an ischemic stroke, and these kids need aspirin. What we see here, in my eyes at least, and I think it's very much what you said, Paul, is vascular fragility that we really don't completely understand. It may be because of the surrounding cells, it may be because of the endothelial cells themselves, that lead to hemorrhagic strokes for the most part. I've not seen any data showing that there is true vascular wall inflammation with gadolinium enhancement and the whole 9 yards, primarily resulting in the stroke. So we have to be careful when we're saying strokes equals no aspirin equals ADA2 deficiency. I think it's a subgroup, but it's distinctly different from other childhood strokes.
Yeah, and Susa, this also raises the question, if one hypothesizes that some patients may have small vascular fragility kind of events, that those may be quite asymptomatic. It may be the sort of thing that one would not easily be able to detect, certainly by scanning, and maybe not by other kinds of testing as well. And it then raises the question, which we're going to be discussing a little bit later, about the whole issue of treatment or not of quote-unquote asymptomatic individuals. How sure are you that they're asymptomatic?
So when you showed us the pictures before you published the paper, you came to Toronto and showed us the histology of the brain tissues. And we have about 100 brain biopsies of kids with small vessel vasculitis, and none of them looks like the ADA2 picture. It's these are vessel wall inflammation with surrounding and intramural infiltrates. So it is a different pathology. I'm not saying we shouldn't look for it, and it may not reflect a distinct phenotype within. But I don't think we should just label them strokes, because childhood strokes, for the most part, are different from ADA2 strokes. And you're right with these could be microhemorrhages, and that's what I said when you showed me the pictures.
Yeah, and so for those of you who aren't familiar with what Susa is talking about, we published in the New England Journal article a couple of years ago some histologic images from the brains of patients. Now, we don't routinely do brain biopsies on our patients, so I don't want you to think that that's what's going on. These were a couple of patients that had hemorrhagic strokes where, in fact, they had to have their hematoma evacuated, and in the process did have a brain biopsy, and that's essentially what Susa is referring to, is the histologic examination of that tissue. Yes?
Quick question for you. Have you sequenced the KECR1 gene and excluded heterozygous mutations in all of your patients?
No, but if you're offering.
I'm sure there's someone in the room, because 150 individuals sequencing for this rather short gene is these days very well possible.
So I mean, this is not a joke. I think we're at a stage where many of you in the room have characterized what we consider a rare disease, primary CNS vasculitis, and I strongly believe it's a monogenic disease. We have to figure out what kind of gene it is, and whoever is keen on doing it, we have a wonderful cohort. I think we can learn a ton, yes.
All right, well, thank you very much. I think this has been a great discussion, and maybe we should move on to another question. The second question is, in terms of screening, is activity level or sequencing the best first test? Which is most economical? So who would like to field that question?
We kind of do both of these tests. I mean, we started really doing sequencing because it's relatively cheap, easy, it's not a big gene, so it's easy to test. However, once we encountered these patients with single mutation, we really had to consider also a protein-based test. So at this point, I should not say that we run routinely on all these patients, both sequencing and also protein-based tests. But if needed, yes, we ask for serum samples to be sent to us. And it's a kit it's commercial kit, basically, It's not certainly CLIA approved, clinical testing, but, we use it just to confirm the clinical diagnosis of DADA2. Whether we should go in that direction, I don't know, it depends what is available to people and whether they can contact Mike and other people who can run all these tests and so forth. Sometimes, we cannot do certainly testing from dry blood spots, right? Mike can. So for us, in order to get serum sample or plasma sample, it has to be shipped on dry ice, which could be a problem, you know, for some of the physicians to send the sample to us and so forth. I mean, I would still think we should go first with sequencing and then complement with protein assay. So,
well, the story I get from people who contact us is that they suspect that it might be ADO2 deficiency, and it's going to take a while to get the results back from sequencing. And would we do enzyme assay? And we don't, you know, we try to get the result back very quickly. So our screening assay, if we find that the patient looks deficient by that, we can just call you up or send you an email, even though we won't send a final report until we do the HPLC assay, which takes another day or two. We don't charge. So, you know, in terms of, we could easily get overextended, but we haven't. And, you know, so we're happy to continue to test. I can't say what the best first test is, because one problem is that we don't know, or we don't have a systematic way of knowing what the genotype is of the patients we find to be deficient or not deficient, because we don't have we never set up any kind of mechanism for getting that information later on once the genotype is known. All I can say is in almost all cases where the patient's deficient, we've heard that the patient's genotype is abnormal. You know, they do have 2 mutations or 1 and 1 suspected. So I don't know if I can answer that question, but doing the enzyme assay, I think, is reliable, and soon we will finish our validation, and then we'll have CLIA certification for it, for what that's worth. And, you know, we're happy to do it.
I mean, it's just a matter of, you know, what you are used to do, right? So, you know, I honestly don't feel comfortable with this assay that we run in our lab, because we are not really a biochemical lab, and I always worried, you know, whether we are overestimating, underestimating some results or whatever, although I show the data, which seems to be okay. I mean, but I feel more comfortable just because I spent so many years working with sequences, you know, for me this is easier thing to do. I feel more comfortable saying someone has ADO2 deficiency based on the genotype than on the assay that we run in our lab. But if you run a biochemical lab and you are comfortable with assay and so forth, it makes a lot of sense. And I believe that you can get results sooner than we can get them, you know, by sequencing and so forth. So that's certainly the advantage. So
maybe this is a good place just to say one thing. With ADA1 deficiency, and, you know, we've tested over 2,300 patients over the last 20, 25 years and diagnosed maybe 300. One difference between ADA1 and ADA2 deficiency is that in ADA1 deficiency, you have a very powerful biomarker, which is elevation of deoxy-ATP in red cells. And we have no biomarker other than enzyme assay in plasma for ADA2 deficiency. And I would urge anyone who has associates who will not only do mass spec, but will do unbiased mass spec, looking for something other than something they already know what it is. Because I think we can always, with ADA1 deficiency, even if the patient's been transfused and have red cell ADA activity, be very confident that if the ATP is elevated, the patient is very likely to be ADA deficiency. And then we do our own sequencing of genes to confirm the biochemical diagnosis there. We do not want to get involved with ADA2 gene sequencing, because that can be done by people who are really good at it. And
so maybe a couple of practical points that I would raise with regard to this. And those are, and they sort of came out in Mike's and Ivona's answers, and they have to do just with the speed with which any particular investigator or clinician might be able to get an answer from a reference laboratory. There is the issue of, if this is going to be done on any sort of a larger scale, that perhaps CLIA certification is going to have to, at least in the United States, is going to have to be implemented in order to have some sort of a reliable scheme in terms of testing. And of course, one of the questions that I heard from Troy is going to come up in a little bit, but maybe we should talk about it now, is, so in terms of screening, you know, which is the best test, but there's also the question of who should be screened. And, you know, we've certainly heard during the course of the morning session a number of different phenotypes that may be associated with mutations in CECR1, and, you know, including unexplained stroke and polyarteritis nodosa and liver disease and immunodeficiency and so forth. And so one would have to consider what is the denominator that we're talking about. You know, what is the universe of individuals that should be screened? And even, you know, one could, based on the very interesting and detailed account that Yaakov gave, maybe one would want to do it at the newborn level, because some of those children in Israel presented at a very early age. And if it were practical enough, maybe we would want to add that to a screen. Yeah, Allison.
I just want to, so one of the challenges that I've recently encountered is the question of so I prefer the activity assay first, right? Because if the activity is normal, what we find in terms of DNA is not going to be influencing our clinical decision making, right? So, you know, my preference would be to get the activity test. However, right now, what I've been faced with is that's a research test. Do you have your patient enrolled on on a research study? And that's where we've run into some difficulties. And so our approach now is to have them on at least some study, maybe not yours, but also in addition, do the sequencing at the same time. The other challenge, again, you can't just order CECR1 sequencing alone. You know, it's going to be part of panels or exome, and that's very expensive. So I think the activity assay is really where we need to go, but to be able to do it in ways that, you know, is appropriate clinical medicine, it needs to get at the point where it's through these appropriate channels and not in a research setting.
Paul, you wanted to say something?
I definitely agree with that. You know, as a show of hands, I'd really like to ask how many of your patients have been diagnosed from anything other than research. I suspect that there's not a single one of the tests that we're doing that really are routine tests that a physician can click on a computer, get a result back from you know, maybe with the exception of the genetic screening that you offer.
Sequencing you can, yeah, for sure.
So, so not in every country. So, for instance, in the UK, it's only offered as part of research programs at the moment. It has not yet filtered through to routine practice. It needs to, and that's definitely something that the Dada2 Foundation should push and advocate for. The second point is I agree with you, because at face value there's going to be probably no patient with confirmed genetic Dada2 who's got a normal ADA2 enzyme activity, but there are some problems, because in the vasculitis world we do things to our patients that might confound the result. So if I have a sick patient who's had a terrible stroke, who may have other things, I may have given them some IVIG which contains ADA2, or I may have undertaken plasma exchange, which is someone else's plasma. So if you're doing a mass screening based on that strategy, you need to be mindful.
That's a very good point.
Dan?
Yeah,
Dan?
please.
Dan? There's several questions.
Okay, yeah.
One more comment about the screening. So I was wondering, of course, if you do genetic screening first, you might pick up all those polymorphisms, and we don't know about it. But I was wondering if we are actually missing out on patients if we do only the enzymatic activity, because I know that we had this discussion before about heterozygous patients that turn out to be intermediate in their activity, but they have a phenotype of vasculitis, they have immunodeficiency, they seem to have the disease. We can only find one mutation, we can only find intermediate levels of enzymatic activity. So what do we do with those patients?
Yeah, they're very interesting. And you know, related to this is the question of whether the enzymatic activity is crucial, really, for the function of ADA2. And Andrei Zavialov, I hope, will talk about that. So it's conceivable that there are patients like the one that we've talked about, who has one known mutation in one allele and has either a polymorphism or a variant of unknown significance in the other allele, who looks like ADA2 deficiency clinically, but who has plenty of ADA2 activity and maybe elevated compared to at least our healthy controls. Well, could that patient have a mutation in the second allele that affects the function of ADA2 but not the enzyme activity? And that, I think, is a very at this point, we really don't know the answer to that, and I think expressing whatever that second allele is carrying would be important.
So we do have some comments from the audience here, so why don't we start?
Yeah, just briefly, this would be for Mike. So the old newborn screening test that was done in New York State in the dark ages for ADA1 deficiency, could that be adapted for ADA2 with just adding ENA to it?
Well, that didn't even work for ADA1 deficiency, and the problem is it was based on release of ammonia.
Right.
But without going into the technical details, over a several-year period, they missed some cases of authentic ADA1 deficiency and picked up several individuals with partial ADA deficiency who were healthy. But I don't know about newborn screening for this disease. I don't think we have a treatment other than one that is easily available at the time that they present, which is anti-TNF.
Yeah, I wasn't necessarily.
I don't know that we know when the disease is going to be expressed, even in families where one sibling may express pure red cell aplasia at a near birth or as an infant, and yet have an asymptomatic sibling who won't express the disease until they're an adolescent. So I think we're right now in conversation with a group that feels that there's a good reason, I agree with that, for doing prenatal testing in a pregnancy that's at risk, and there are issues about how to do that.
Yeah, so one question that's related to the ideas that you're bringing up, Mike, is that we really don't know, and this sort of comes back to the question that Chip was addressing at the very outset of the meeting. What is the frequency of individuals who have predicted deleterious mutations in this gene? We don't know in the general population. And actually, that is something that could be learned by looking at large databases of sequence, which are now being generated. And it would be very important for us to have some idea as to, what is the extent of this?
You can mention this number then that we discussed yesterday. So basically, Pearl or Qingzhao and I, we look at, or sort of hypothesized, what would be expected incidence of this disease based on the frequency of pathogenic mutations in this gene, and we estimated that there are likely, this is again estimate, 4 in 100,000, something like that, which is a rare disease. I totally agree with you, but still there are a lot of patients, and again, if you just rely on family studies, as you mentioned, because mother or father is affected, or I don't know, there is a disease in a particular family, we are going to miss a lot of patients, like were the 3 first patients that we involved in our study. I mean, they were sporadic cases in the families, and
but
that's
4
going to
on
happen.
100,000, so just to do the calculation, so 4 on 100,000 then is going to be 40 per million, and if there are 300 million people in the United States, there's more than that actually, then that's going to be 12,000 cases in the United States.
So I think we have an opportunity to answer this question. I'm Philip Reyes. I'm at the University of North Carolina, and I've been asked to help with a research protocol for newborn screening doing whole exome sequencing. And Mike, I was going to ask you if you wanted to help argue that we start screening for ADA2 on that research protocol.
I'm sorry, I don't have a research protocol.
No, but I'm being asked to give them list of candidate genes of places we could step in early on, because they're doing this more as an ethical study to look at the ethics behind screening, but then they've asked clinicians who take care of diseases that might be picked up in newborn screening that you would actually intervene, like say HLH as a Cincinnati alum. And so I think we have an opportunity, if we have more backing and expertise, to guide them to look at their sequencing data, to look at the frequency, at least in the state of North Carolina.
I'm not sure if that question was for me.
It's not really, if you guys are willing to,
Yeah, I
to,
think-
to help push for this, I can.
I'd be happy to help. I'm not sure, do you mean by assaying enzyme activity in?
So we could ask them to look, to do the sequencing at the newborn period, and if we find, mutations, then target those patients to come in and look at levels.
Oh, we'd be happy to do that. I mean, I would have to get an IRB protocol
Correct.
to do that.
Correct.
I have to say that we don't have an IRB protocol, and we have kind of a waiver to do this as standard of care testing
Gotcha.
when a physician asks us to do it, not as part of a study where we go out and ask people to send us samples. So I know
So we'll talk offline.
yeah, we'd have to talk about that, but I have only, the whole, yeah, definitely do the screening if you can get,
Okay.
if you have a reason to do it, do it.
Panina, you?
I would like to add a short comment relating to our population in Israel of Adatu that are mainly Georgian Jews. I think that in such a population, maybe some screening like marital advice, marital counseling could be really effective. And actually, mother of two kids with Adatu made it for her daughter, non-professionally, but very well. There was a guy who was not Jewish Georgian who courted her daughter. And all the family were against this because he wasn't a real Georgian Jew. And he was from Kurdistan. But the mother said, and she was a strong woman, that no, I want him to marry her. And actually the couple is here as part of the patient's meeting. And they have two children that we didn't try even to measure the ADA2 or to sequence because the father is Kurdish. I hope there is not something in the intron that will show itself later on or something like this.
Thank you, Panina. Gigi.
So mine is a comment actually to a very important statement that Mike has just made, and I would like to bring it to a next level. So he actually pointed out that there would be a need for some unbiased approach, maybe mass spec, maybe some others, trying to identify biomarkers of this disease, and that maybe might facilitate the diagnostic process. I think this is not just finding biomarkers, but we do need unbiased approaches to identify what is actually the function of this enzyme. Is it truly an adenosine deaminase, or is it mostly an adenosine deaminase? Does it have additional targets? By using an unbiased approach, we might be able to identify a signature that tells us more about the pathophysiology of this disease, and we are very much in need of this.
Yes, Gigi, I totally agree with you, and really, in order for us to be able to do appropriate screening, we have to understand better what's really going on in this disease. And although we have maybe a rudimentary idea as to what's going on in this disease, it really, at this point, is early days. And just to put it in perspective, and I don't want to be discouraging in what I'm about to say, but the gene for FMF was identified in 1997, as has been mentioned before, and we're only now beginning to understand what is the normal function of pyrin, the protein that is mutated in FMF, in 2016, 19 years later. And there's reasons for that. It is a difficult gene to work with, but still, it underscores the fact that there have to be really concerted efforts to understand more, what is the biology of this disease. And some of that may be studies of cohorts of patients, some of that may be development of animal models, some of that may be various kinds of screens that one can do in vitro assays, but more needs to be known in order for us to be confident as to who really should be screened and what should they be screened for, too. So I think that's an important issue.
Can I ask just maybe one last question to wrap this up? So Mike, in terms of the enzyme screening assay, are there any patients with ADA deficiency, in other words, symptoms, who've had normal enzyme activity? I guess, how sensitive is that screen for detecting patients, for those of us seeing patients in the clinic sort of as they come in? How comfortable can we be with the enzymatic activity being sensitive enough to pick up the majority of those patients? Not sort of the oddball ones, but...
ADA2 deficiency.
Yes, the enzymatic assay. Have you had any normal levels in patients subsequently found to have pathogenic mutations, I guess is my question.
Not that I'm aware of, but that's the problem. The way we got started with this is just by word of mouth and people sending us samples. It wasn't that we were measuring the activity all along and we knew anything about the range. We've learned something about the ranges in the controls that we've been sent, which we're not sent the age of the controls, but from having asked around, they're adults. What Paul said is very true, and we've asked to get pediatric samples for controls, but I'm not in the clinic. I don't have children of my own. I can just take their blood or something. But that's a very important.
Yeah, and I'm not sure I entirely agree with the carriers having an intermediate level because, in fact, when you look at them compared to adult controls, they're no different.
Well, no, in our hands, they're very different. They're statistically significant, but I've forgotten the number on the slide that we've done, but I think it's around 40 healthy adult that people have sent to us. And compared with the carriers, who are genotypically confirmed as carriers, it's statistically significant. Now, that's the population that we've studied. We haven't studied hundreds and hundreds.
And the question is, how many are adult age 20? How many are adult age 60, age 80?
Very, very, I accept that criticism exactly. You are absolutely right. And, we're happy to do it, but we need to get the,
Can I say one thing really quickly?
Oh, yeah, Mandy.
I just want to say we have one patient who, if you just screened his level, he could either kind of be towards the bottom of a heterozygote state or maybe the very top of the affecteds, but he had 11 strokes, so clearly he was, and so I think that would be one of those gray areas where maybe if you just got the levels, you wouldn't want to rule it out just because it seemed to be maybe about heterozygote state.
Yeah, let me say that's one reason why we set up 2 assays for confirmation, and because we have more confidence in low values that we get with the HPLC assay. So if you want to send us a sample on that patient, we'd be happy to do it with the caveat that, as Paul said, we don't know the ages of our controls. So there's going to be some uncertainty, but at least it would be another lab that would test it. And I think, Allison, you asked me whether we share authentic samples with other clinical labs. In one of your, you sent me an email. Yeah. And we would be happy to do that, but you should all understand that there is no standard for ADA2. That's why we made the recombinant protein. And, so it would be great if a clinical lab, a commercial lab, would get into this and really, several commercial labs. The history of testing for ADA1 deficiency by enzyme assay was that after the disease was discovered in the early 1970s, there were many academic and hospital labs and commercial labs that would do testing for ADA1. It's a straightforward thing. But during the 80s and periods when financial crunches came down and bottom lines became very important, one after another, those commercial labs and academic labs, some of whom were very heavily involved in testing for ADA SCID, stopped doing it. And a few have continued, Baylor, Mayo's. None of them do DATP measures in the United States. So the likelihood is that unless there is some real push, it's going to be hard to get in the United States commercial labs to pick this up.
Jenna, you had.
Just one quick last comment on the ADA2 level. When the ADA2 activity is totally normal, I think it's a question of the definition also of the disease, because you asked how sensitive is this, because for us I can only say that if the patient has a totally normal ADA2 activity, we decide that this patient is not ADA2 deficient, and we stop screening. We don't do genetics when the patient has totally normal levels. I don't know how the others do this, but I think we cannot really say anything about sensitivity because of that.
Mario, you have
just a quick one. Yes, Gigi and I were talking about, and I can't resist asking the question. Has anyone seen low or unmeasurable ADA2 levels in patients or in people samples with no mutation?
With no mutations?
Yeah, otherwise in any other
I think just in one patient with a complete phenotype that was negative, let's say negative, but has a complete phenotype in terms of strokes, hypogammaglobulinemia, and livedo, and so on. It was affected.
Yes, and had low ADA2 levels,
yes,
right, or
yes.
not measurable. But the story about this patient, Marco, is that this is a patient from Sardinia, right? I remember that. So you can think again about, founder population in Sardinia, right? And we mentioned these copy number variations, deletions, right? You are not going to see them in patients referred to us from American population. Very unlikely that patient will be homozygous for copy number variation, some deletion. But you are more likely to see patients like that, again, being homozygous for the genomic deletion that you will not be able to see, by doing just regular Sanger sequencing. So my point is, obviously I feel more comfortable doing sequencing than, these protein assays. I don't dare to interpret data tool based on the protein assay that we run in our lab. To me, this is just sort of complementary, So I like to have both, information in order to call the clinical, to say that someone has a data tool, basically. But maybe, with years, some biomarkers will be developed or better assays or something that, would be easier to do testing.
Yeah, there's one other thing. Again, I don't know if this is the best place to bring it up, but there is the possibility that ADA2 functions by binding to some receptor. And again, I hope we'll hear more of that from Dr. Zavialov. But so it's conceivable that there are phenocopies that will turn out to have normal ADA2 levels and no mutations in CECR1 who have another gene with which it interacts that is mutated. And hopefully with more and more sequencing, that will become.
Yeah, maybe we should move on to some other questions here. So at the top of the screen now, at least is suggested, the question, the ADA2 elevation of at-risk individuals, is this in inflamed patients due to what? Bacterial, viral, autoimmune inflammation? I ask as it may be a marker of discrimination, like procalcitonin. So would someone like to address that question? So I think that Mike had seen elevation of ADA2 levels in some individuals that you had described as at risk. And so I think that it's addressing that observation, I believe.
Yeah. Well, I have the age of all of those patients, and I have been trying to find time before I came here, having read Paul's paper, to be sure that it wasn't just due to that they were younger. And I don't think so, because we have patients a whole range of ages, and I'll look at that. But in that case, I don't really know what the clinical history of these patients is. And sometimes I get a little bit of information when I get requested to do the testing. Johanna, you probably know, and everyone else who sent samples to me, the answer to that question, and maybe you can address whether it's, I've always presumed they have some clinical picture that looks like an autoinflammatory disease or some other presentation associated with ADA2 deficiency. And so something, if they're not ADA2 deficient, then there is something ongoing in that patient that is responsible for the higher levels of ADA2 that we're seeing. And it is known that ADA2 deficiency, or rather ADA2 levels are higher in serous fluids or pleural effusion of patients with certain infections, so that's a reasonable possibility.
I think that specific patient that you're referring to it's a little bit difficult because actually it's not such a classical ADA2 phenotype. That's why we were also not so sure about it. It was more like CVID and lymphoproliferation, not so much inflammation, and that's why we were also like hesitant about those results. And that was the only patient I know of with the elevated levels.
A related question is why do patients with ADA2 deficiency who can go for years with no symptoms suddenly have a catastrophic neurologic event? What is the provoking event? Yaakov, you want to speak to that?
Several our small kids presented after some immunological stimuli like vaccination. Similar things are happening in hyper-IgD syndrome, where the first attack is provoked by some inflammatory trigger.
Yeah, and in fact, the Georgian Jewish population might even be, because of the relatively high carrier frequency, a population that would be good to study prospectively to get some sort of an idea as to what the risk is of being a heterozygous carrier, because there's a high carrier frequency in that population, and what might be provoking events. Yeah, Marco?
Yeah, I think that is rather interesting because there are some patients that have a chronic inflammation. They have a sort of chronic inflammation, and for those it's easy to think that we have to treat them at once as soon as you have the suspicion and so on. The tricky ones are those that have this kind of remitting phase in which they have a long moment. Our experience is that when they have systemic flare of inflammation, they usually have also the strokes, the complication and so on. So that came to the point, there is a question to Paul that I think is very interesting. Treat or not to treat the asymptomatic patient, how you define the asymptomatic patient. I think that this is, in my opinion, I thought, okay, if I start to see some inflammation, for sure I start treating. But now I'm a little bit concerned about that, especially when you say that there are some patients that present strokes even without any inflammation, and after seeing patients with asymptomatic strokes. So sometimes, retrospectively, we saw, I think that is a common experience, some patient that has asymptomatic strokes. And they were asymptomatic also from the inflammatory point of view. So I'm start thinking that maybe it would be worth starting treatment very early. But this is really...
Well, these are definitely matters of judgment and at least to some extent personal preference. And given the fact that we don't have the full set of information really to help us to make those kinds of decisions, it's hard. I'm sorry, Janet.
Okay, I could have an example. At least in our family with a stroke phenotype and a long medical history that was very particularly recorded. Each time before they had the stroke, there was a bacterial infection.
A bacterial infection.
Bacterial infection that was accurately treated, but still after that. So it might be a urinary tract infection, respiratory infection, or periodontal infection, but each three of them always had the infection kind of prior to their stroke.
So it sounds like there's at least a growing body of anecdotal evidence anyway, connecting immunologic stimulation with possible exacerbation. Yeah. Okay. Oh, yeah, I'm sorry. Nora.
So along those lines, there's a long rheumatologic literature on PAN in children following strep infection and PAN in adults following hepatitis B. Has anybody looked functionally to see if ADA2 changes in those viral infections?
That's a great question. It looks like at least our panel of experts here is left scratching their heads. Seiza.
Yeah, hi. All the speakers were great. Thank you. I have something to add about these asymptomatic patients. Now, in our center, we have collected pediatric plus adult patients, which, so these 15 patients will be presented at the ACR. But one of them, who was 20-something years old, had his first symptoms at the age of 20 plus, and he presented with renal amyloidosis, suggesting that he did have an ongoing inflammation, which wasn't, with apparent, phenotype, but he had the subclinical inflammation going on. So, and there was another patient who had his first symptoms at 35. So it looks like, we don't know if these asymptomatic patients are really asymptomatic, and we don't know how to define asymptomatic. And while I have the microphone, can I comment on one thing as well? In these monogenic autoinflammatory diseases we know till now, rarely we see such phenotypic variation according to geography. For example, it's not just the Georgians, but it looks like the Eastern Mediterraneans have this typical phenotype, pan-like, few strokes, because in our series it's only 12% the strokes. Not so much immune deficiency, it's only like low IgM levels are just like 1/3, and we routinely check all of them. So it looks like, and then we see that there are more strokes in the North American population than we heard so much about this immunodeficiency in the German populations. So it looks like this monogenic disease is kind of more challenging than like the other ones, because, for example, FMF, the phenotype doesn't differ so much with the geography, the severity does, or CAPS, or so on. So I find this interesting, and I don't know if you have anything further to comment on this.
Does anyone have any comments about
Dan,
that?
Can I just, I think one of my questions says, is it selection bias, though? Because you're a rheumatologist, you're getting autoinflammatory patients, and that's the phenotype that you're screening, whereas Bodo has this big cohort of CVID patients. And my question is, if you took CVID patients in Turkey and did the same screening, would you find the same incidence as Bodo? I think at this point, yeah, it's not clear to me that this isn't just selection bias.
Yeah, well, that's true. We're now trying to, we're going to start to screen these early onset strokes now. So that is true, though our immunology department usually checks for everything when they have an immunodeficiency patient, but they still might be missing some. But for example, when we screened our PAM patients, what we call PAM patients, and we were kind of sure that they had PAM, more than 1/3 had this mutation, which looks like it's more increased in our part of the world. I think there might be some bias in the
certainly, just given the frequency of the G47R mutation in the Georgian Jewish population, and probably that extends to neighboring areas, if there's a genotype-phenotype correlation with that mutation, then perhaps that could account for at least some of the phenotypic distribution effects. Panina?
Well, one short one and one longer. The short one is that one of our patients had a severe exacerbation post-streptococcal infection. So it's not much to say, but any kind of inflammation may cause this, flare of disease. But I want to comment about something else, which seems to me the cardinal question, or one of the cardinal questions of this meeting, and this is how to deal with genetic mutations that are asymptomatic, and should we give them right away the prevention for life by anti-TNF, or should we wait and follow and decide this and that? We can't make many decisions from our 30 patients, but some comments I think may reflect something. There was one lady who at the age of 60 developed ulcers, and she never had anything, but she had mutations, the Georgian mutation, the homozygous. And again, it's not being pre-symptomatic It's not just one step, pre-symptomatic. I think there are several steps, because few patients that had only LR didn't then develop anything in the last 20, 30, 40 years, several. But 2 patients at least that I can recall did have LR, livedo reticularis, but also mild anemia and high ESR and CRP. And this girl, who was for 12 years asymptomatic and just followed with her brother who was very symptomatic, developed at age of 15 most severe mesenteric vasculitis. And she presents also another problem. I just reminded that she was on Enbrel, doing very well, studying, marrying, bringing up, delivering 2 children, but she found out that after the first delivery, she was doing quite well without Enbrel during part of the pregnancy, because there are rules how to use pregnancy. And only preparing to this meeting, she told me that she's not taking Enbrel in the last 3 years. And so far, she's good, but she had a very severe event of mesenteric vasculitis that almost needed a stent.
Yes, it's a very difficult thing to predict, and unfortunately, the observation of these patients, at least sometimes, unless we have some tip-off as to when a severe event is going to happen, sometimes you're left sitting there observing a catastrophic neurologic event as the first thing that you see. And that, of course, is something that none of us want to be responsible for. Do others have comments about that?
I mean, I think this is the whole ethical issue here, actually. So by asymptomatic, patients may not complain of any symptoms, but they may have a C-reactive protein which is 30, 40, or 50. I would not regard that patient, in my view, as someone who's asymptomatic. I think they've earned their treatment at that point. A truly asymptomatic patient may have absolutely no symptoms and normal inflammatory markers, and I think you should leave those patients alone. You should warn them that things could happen, so if they develop an infection or some other unusual symptom, to seek medical help very quickly. And of course, that does not preclude the fact that they might have an irreversible catastrophic event, but the risk seems somewhat lower. Coming back to Cesar's point, I would strongly suspect that the patient who is asymptomatic, who got renal amyloidosis, would have had levels of inflammation. Had they been monitored, would have been detected, and this may have been preventable using that type of approach. I'm really interested in the fact that you give all your patients anti-TNF.
Yes, we do, and actually there is one who has declined taking it right now because he feels good, and he is one where we've only identified a single mutation, yet he has a sister with a history of 2 strokes, so you get very nervous. And I think because we have such a stroke predominant cohort, it's very hard to go outside of that perspective, because that can be so life-altering. And so we tend to perhaps over-treat because of our experiences with the strokes. Maybe if we saw more of a different flavor of data too, we would go about this differently. But having seen that our most profoundly affected patient went for almost a decade really without having major clinical problems from her disease, and then at age 22 had a massive cerebral hemorrhage, it makes it very hard to put that out of your mind when you see a very healthy child.
I think that this also raises, and then I'll let Marco comment, the question which Paul raised of what is appropriate follow-up on these individuals who have 2 documented mutations, and at least at the time that we see them in the clinic or the office or whatever, they are asymptomatic and have a normal SED rate and CRP. So then how often do you see them, and how often do you monitor their SED rate and CRP, or are there other things that we should be monitoring? Marco?
No, I think that, I mean, of course, it's a relevant question, and sometimes we can miss some inflammation in between the visits. So I mean, it would be very important to check very carefully, especially these patients that are really tricky. My opinion is that after this fantastic meeting, we have to choose to make a point on the fact that it would be really important to have a sort of longitudinal registry for these patients. This is the only way to respond, to collect the few patients that we have, the few patients that we have in different centers, but they are a large number of patients now, and to follow them prospectively. That is the only way to solve it, to reply to some.
Actually, I have a question. We're focusing on these individuals with known mutations but haven't had any significant clinical features. I'm just curious, how many patients are out there that meet that definition? I mean, I'm just wondering if, like a show of hands, how many people have identified, and I assume it would be through family studies, individuals who have biallelic mutations but don't have any phenotype at all. So maybe 1, 2, it seems like they're pretty rare. Would that be a true statement? Like nothing, well, okay, so let's, yeah, nothing. I mean, the ones that you would define as pre-symptomatic, how many, because we're talking a lot about them, and I'm just curious, how many of those are there out there?
I've got 8.
Okay, that's a good number.
Yeah.
And their age range? In their ages, I'm just curious.
Okay, so I call them, so there's 2 filters before you get to get treatment. Okay, there's us pediatricians, and then there's these things called adult doctors. And one of the consanguineous families, they're adults, so I send them to Philip Hawkins and Helen Lackman, who laugh at me at the prospect of treating a well individual in their 40s with a normal serum amyloid A who's got maybe a bit of carpal tunnel, but it's probably carpal tunnel and nothing else. And yet they've got 2 copies of the mutation. Their children have got the phenotype. They've got absent enzyme activity.
Yeah.
I have a question.
So they have absent enzyme activity.
Yeah, all of our patients read that with mutation plus
right,
enzyme.
Right. The only thing I'd wonder is whether, if you looked at their monocytes for secretion of the enzyme, whether you might get a surprise. And I only mention that based on the pretty well-established occurrence, or fairly not infrequent occurrence, of somatic reversion in lymphoid cells of patients with ADA1 deficiency, where Troy knows some of these patients, you know, where when you look at their lymphocytes, you see a skewed population at the time of diagnosis. They might have a high level of CD8 cells. They turn out to have had one allele that's reverted. And so I'm wondering if in patients like this, whose serum level is very low, there may still be a population of monocytes in which there's been a reversion of one allele enough to, you know, compensate.
Yeah, we have a question
I think
over
going
here.
along with that, my question is also to screen. Like we see these patients in the clinic, some patients that we think they could be in the spectrum because the spectrum is so wide, and who to screen, because what are you going to do if you know that this patient is positive? So I think with some guidelines, hope we can come up with what are the criteria to screen these patients, patients to start with, not waiting for an index patient or so on, just we see someone in the clinic with deep livedo reticularis or ecchymosa and maybe Hashimoto's thyroiditis.
Does anyone want to comment on that?
So what we're getting into here is outside of data too. It's the mathematics and ethics of screening. So if you have a pre-test probability that's high, in other words, you've had 11 strokes, you find a borderline A to 2 level, that's relevant to you because your pre-test probability is very high and it just goes a bit higher. But if you're a completely asymptomatic individual from an outbred population and you do mass population screening and you find a borderline A to 2 level, it's probably irrelevant. And this is true of every screening test that's ever been invented. But even before you get to that point, you have to ask yourself, well, if you do find, as you mentioned, an abnormal result, what are you going to do about it? And as you can hear, there's a difference ethically about doing that approach because you may not have any treatment to offer them at that point, and therefore you convert the burden of unknowing, or no burden at all because they're into population screening, to a severe burden of knowing. Yeah?
So one way that one could, at least at the genetic level, do this without any additional cost would be simply to take data that are already being generated in terms of exome sequences or genome sequences in various countries where, either in the UK or the US where these things are being done, in other countries I'm sure too, and get some sort of an idea as to what is the frequency of mutations and biallelic mutations in those populations. And then perhaps, at least for those who have consented to recontact and follow up, to get some longitudinal information with regard to what happens to those individuals. Okay,
the
now
frequency of biallelic mutation is going to be high because this gene is so rich. But how do you decide what is the pathogenic mutation? That's another question.
One may have to
right. So that's another level of analysis, right? Because it depends what are you going after. What type of mutation and so forth. So.
Okay, well, Troy is signaling to me that the carriage is turned into a pumpkin, I guess, is one way of putting it. And so I think that at this point we're going to have to call this session to a close. I'd like to thank all of the panelists and the audience for a wonderful discussion.